Special focus is in ophthalmology. Genetic defects in over 200 known genes have been associated with poor visual function. Genes are segments of DNA that direct the cell to produce proteins that perform a vast array of functions in the body. Many diseases have a genetic basis, meaning that mutated genes provide incorrect instructions that cause the cell to produce an abnormal protein, or disable production of a protein completely, either of which can cause disease. In gene therapy, a functional copy of the gene is delivered into a patient's own cells. Normal protein that is produced from a functional gene has the potential to correct the underlying cause of the disease and provide restorative therapeutic benefit. Gene therapy uses engineered viruses to deliver genes into cells. These are called viral vectors. Vectors made from adeno-associated virus, or AAV, are especially well suited for treating retinal diseases. AAV is a small, simple virus that elicits only a weak immune response and has never been shown to cause disease in humans. In gene therapy, the two native AAV genes are removed and replaced with DNA that contains a functional gene with instructions to produce a normal protein. The DNA also contains a promoter, which drives production of the functional protein. The promoter and gene are flanked by signals called inverted terminal repeats that allow the DNA to be packaged inside the protein shell of the AAV vector. AAV vectors are delivered to cells in the retina by routine ophthalmic delivery methods, such as intravitreal or subretinal injection. Once administered, AAV vectors enter the patient's cells and deliver a copy of the functional gene directly into the nucleus. Once inside the nucleus, the functional gene can support the production of the normal protein and correct the underlying cause of the disease. Genetic defects in the eye can cause vision loss or blindness, and gene therapy can provide a long-lasting therapeutic benefit. AGTC is using its technology to develop gene therapies for rare, inherited retinal diseases that currently have no treatments. One of these diseases is achromatopsia, an inherited retinal disease which is present from birth. Achromatopsia results in markedly reduced visual acuity, extreme light sensitivity causing day blindness, and complete loss of color discrimination. The retina is a layer of cells at the back of the eye. In a healthy eye without achromatopsia, special cells in the eye, called cone photoreceptors, convert light into an electrical signal, which is transmitted across the layers of the retina and then to the brain for visual processing. In an eye with achromatopsia, cone photoreceptors in the retina are unable to convert light into an electrical signal. The most common causes are mutations in one of two genes, called CNGB3 and CNGA3. In gene therapy, an AAV vector containing a normal CNGB3 or CNGA3 gene is designed to provide the instructions for normal CNGB3 or CNGA3 protein and restore cone photoreceptor function. By introducing a functional copy of a gene into a patient's cells, gene therapy is able to correct the underlying genetic defect that is causing disease. The functional gene, delivered by an AAV vector, is thought to remain stable for many years, which suggests that gene therapy has the potential for long-lasting clinical benefits after a one-time administration of the vector.
Many human diseases are caused by defective genes and improper protein functioning. Genes within our DNA provide the blueprint for the production of proteins. Specific gene defects or mutations can result in reduced or inability to produce a functional protein or production of a protein with altered function. Gene transfer using adeno-associated virus, or AAV, vectors can target specific cells to correct disease-causing improper protein function. Once inside the target cell, therapeutic genes within the AAV can correct defective or missing protein function. To achieve this, AAV genetic material is replaced with a therapeutic gene to restore natural function of the disrupted proteins. A protein shell, called a capsid, encases the genetic material and helps target delivery to certain cells. Once within the target cells, the capsid is shed inside the nucleus to release its vector genome. The vector genome does not integrate into the nuclear DNA, but instead forms an episome. Episomes are not passed on to future generations of cells, thereby ensuring that the effect of the gene transfer is not passed on. Using the body's natural machinery, this allows stable and long-term expression of the therapeutic molecule. AAV-based vector delivery systems can be administered by intravenous drip or direct injection to target tissues. AAVs are ideal vectors for gene transfer, as they are non-pathogenic and capable of transducing both dividing and quiescent cells. The different components of AAV-based gene transfer can be switched to address different therapeutic targets and diseases, and several studies and clinical trials have indicated that there is activity in different disease models. Gene transfer offers the potential of long-term and potentially curative benefit to patients with genetic or acquired diseases by correcting defective or missing protein function through a single administration.
In the pursuit of a cure for AIDS, scientists have devoted great efforts into studying the HIV lentivirus. Although a cure for AIDS remains to be seen, research efforts into the HIV lentivirus have led to major developments of powerful HIV-based tools for transgene delivery. Today, lentiviral vectors are major tools for basic and translational research, enabling scientists to stably overexpress or silence genes, immunize against a range of diseases, generate transgenic organisms, and much more. The lentivirus is part of the retroviruses family of viruses, with HIV-1 being the most widely studied. Members of this family contain a positive single-stranded RNA that is reverse transcribed into DNA and integrated into the host cell's genome. The HIV lentiviral RNA genome contains three major genes, GAG, POL, and ENV. The GAG gene encodes protective core and matrix proteins for viral assembly and infection. The POL gene encodes for reverse transcriptase, RNase H, and integrase, enzymes crucial for reverse transcription and genomic integration. The ENV gene encodes for surface glycoproteins that determine tropism and enable entry into the cell. The lentivirus genome also contains two important regulatory genes, TAT and REV, that activate viral transcription, as well as four accessory genes, VIF, VPR, VPU, and NEF, that are less essential for virus replication in host cells. Lentiviruses have lots of potential as tools for gene delivery and gene therapy. This is because their stable integration into the host cell genome ensures long-term expression for stable cell line generation. They can infect both dividing and non-dividing cells such as hepatocytes and neurons, cells that are notoriously difficult to infect. They have broad tropism, making them effective vehicles for delivering CRISPR sgRNA constructs to edit the genes within any tissue type. They don't generate immunogenic proteins, and they can deliver transgene fragments as large as 9 kb. Because of all these advantages, great efforts have been put into developing recombinant lentiviruses for clinical and research use due to their robust infectionability. However, precautions are needed to ensure replication-competent lentiviruses are not accidentally generated via recombinations between the delivered as well as endogenous viral elements in producer cells. In an effort to reduce biosafety risk, 3 generations of recombinant lentiviral packaging systems have been developed, with the first generation system no longer in use due to biosafety risks. The second generation system splits the essential components of the lentiviral system across 3 plasmids that are delivered separately for safety. The transfer plasmid encodes for the transgene. It also contains cis-acting elements such as the 5' and 3' LTRs essential for promoting RNA polymerase II to begin transcription of viral mRNA, and the size sequence which signals genome packaging. The packaging plasmid is provided in trans and encodes only essential transacting genes, GAG, POL, REV, and TAT, that are required for entry and integration of the viral genome. The envelope plasmid contains genes encoding for envelope proteins. For safety reasons, all other genes involved in viral propagation, for example, VPR, VIF, VPU, and NEF, are excluded. To almost completely eliminate dangerous lentiviral recombination events, an even safer third-generation system was created. In this system, the viral genome is split into 4 plasmids. A plasmid containing only the packaging genes, GAG and POL, a plasmid with only the regulatory gene, REV, a plasmid carrying only the envelope gene, ENV, and a transgene plasmid. In addition, the LTRs flanking the transgene are further modified as they contain enhancer and promoter regions that can activate adjacent cellular proto-oncogenes. By removing this enhancer and promoter region, U3, from the 3' LTR, a replication incompetent, self-inactivating system is created as this deletion is transferred to the 5' LTR after reverse transcription and integration. The U3 of the 5' LTR is also replaced with a CMV promoter to eliminate the need for the transcriptional transactivator, TAT. Although the third generation is significantly safer, its viral yield is typically lower than that of the second generation system. At ABM, we offer both second and third generation lentiviral packaging systems to suit any application. Finally, as tropism is determined by the glycoproteins on the viral surface, a lentivirus's tropism can be further broadened by replacing the HIV-1-ENV glycoprotein with the envelope glycoprotein G from the vesicular stomatitis virus, VSVG. VSVG is incorporated in all ABM lentivirus packaging systems to widen the range of cell types the virus can bind to. Recombinant lentiviral systems show great promise as potent tools for gene therapy to treat HIV infection, cancer, Parkinson's disease, and many more. A breakthrough is surely not far on the horizon. In the meantime, recombinant lentiviral systems have secured their place as an invaluable tool for advancing in vitro and in vivo research across a variety of areas in biology and the life sciences. From siRNA lentivectors to CRISPR-Cas9 lentivectors to chemate-inducible lentivectors, our lentiviral vectors and ready-to-use lentiviruses can be used to manipulate gene expression of any human, mouse, or rat gene. Simply select your gene of interest as well as a promoter, tag, and/or reporter, and we will package it for you at the titer you desire.